Managing coastal resources sustainably through biological sampling. Conducting biological sampling provides information that is essential for effective management of coastal fisheries resources. It is useful in understanding species life cycles and in determining certain demographic parameters such as maturity, growth, and genetic connectivity. This video introduces a series of videos in which you will learn techniques to collect the following samples from coastal finfish: finclip, mussel, otoliths, and gonads. The list is non-exhaustive as many other parts may be sampled to obtain other indicators. Each sample type provides specific information on the species biology, ecology, or its stock status, and can even provide information for public health purposes. We will now describe the information that can be obtained by analyzing these samples. DNA can be extracted and analyzed from a fin clip or piece of mussel. A piece of mussel provides information on the specimen's nutritional composition, contaminant content, and their place in the food chain. Otoliths provide information on the fish's age and growth. Gonads are used to determine sex and stage of maturity. While such samples are relatively easy to collect, certain analyses can be costly and are technically complex. You can extract DNA from a fin clip or a piece of mussel and compare it to the DNA of other specimens to confirm species identification where fish species are hard or impossible to distinguish, determine the genetic structure and connectivity of different subpopulations living in different geographic locations to understand interdependencies within stocks. Mussel tissue can be used to analyze several different components. Isotopes which provide information on fish ecology, particularly their position in the food chain, such as a top predator or herbivore. Contaminants present in the flesh may come from natural or human sources. Studying them can help determine whether local measures on health and safety or consumption habits are needed. Studying mussel composition provides information on the nutritional quality of the flesh that is eaten and is useful in drafting nutritional recommendations. Otoliths provide information on fish age and growth. Otoliths are calcium carbonate structures in the inner ear bones, that the fish uses to position itself in space and perceive sounds and vibrations. Each fish has 3 pairs of otoliths, which vary in shape and size by species. The analysis of the largest pair, the sagitta, reveals the age of the fish, which is determined by counting the clear and dark bands present on a transversal cut, much like reading the rings of a tree trunk. When we know the age and size of several fish of the same species, we can calculate the speed of growth of that species. If we know how long it takes a species to reach a certain size, we can determine whether its growth is fast or slow. Gonads are the reproductive organs of fish. Ovaries are the female gonads that produce oocytes, while testes are the male gonads that produce spermatozoa. The union of oocytes and spermatozoa from fertilized eggs, which will then produce larvae. For some species, the sex is determined at birth and retained throughout their lives. But for others, such as the parrotfish, the sex may change during their lifespan. To avoid any confusion, it is important to be well informed about the species studied before sampling the gonads. It is possible to determine the sex and stage of maturity of a fish with the naked eye. When the size, sex, and stage of maturity of several fish of the same species are known, it is possible to determine the age or size at maturity, that is, the size or age at which this species can reproduce. In the field of fisheries, we refer primarily to the size at which 50% of specimens are mature, known as L50. The lower the L50 value, the earlier in life that species can reproduce. To help determine the sex and stage of maturity, gonads should be studied during spawning periods. If the reproduction periods are unknown, the gonads can be observed on a monthly basis to determine spawning periods. In mature individuals, the gonad size and weight increase as the reproduction peak approaches. By determining the sex of several fish of the same species, we can also calculate the proportion of males and females present in the population. If the ratio of male and female is skewed, which can happen among hermaphroditic species, subject to strong pressure from selective fishing, the species' reproduction success may be compromised. Conclusion. To summarize, biological sampling provides information on various aspects of the biological cycle of fish populations. This information, combined with environmental and fisheries data, enables us to assess stocks and their resilience in response to pressures from fishing and climate change. A fish population is resilient when it can persist in the face of fishing pressure and climate change. This population is all the more resilient if it grows quickly and begins to reproduce early in its life, and if its subpopulations are connected to large geographic scales. A population with low resilience is vulnerable to pressures from fishing and climate change. It will require special management efforts to ensure that this resource is harvested sustainably. So your biological sampling work is important in order to better understand and better manage your coastal resources. In the next episodes, we'll learn to sample fin clips, mussels, otoliths, and gonads.